Okay, welcome back, everyone. Hope you've managed to take in some calories and probably some caffeine and plenty of networking. I've got one job to do before I just hand over to Lisa for the panel, and that's let you know what the result of the vote was before lunch. Was it going to be Massachusetts or was it going to be Golden Triangle?
You have food over here, so everyone uses.
You think? I guess what was surprising, maybe, on balance, was the fact there was only 3 votes in it. It was really close. But Massachusetts won by 3 votes out of that. So well done whoever argued for Massachusetts, which is... yeah. Okay, yeah. I know we're not going for a recount. It was close enough that I think what I thought was great was the fact it brought out that there were advantages and pros and cons in both, and ideally you'd like the best of both worlds, and maybe you can have that as you grow. So without more from me now, it's on to the next panel, and I guess we heard a lot this morning about what diversity in various guises, in business models, in inclusion and what have you. And it means different things to different people. But I guess one of the questions I was left with when I was putting the program together, and this kindly bunch agreed to come and talk about it, and Lisa Melton from Nature Biotech to moderate it, was about whether with all the diversification technology, saying we're taking in different sorts of data and different approaches in biomedical R&D, are the outputs becoming any more diverse? And, you know, it could be either way, good or bad. But over to the panel, and Lisa in particular, to navigate through.
Yeah, thank you, Tony. Welcome, everyone, to this panel on diversity. Just so you prepare yourselves with questions, I'll be opening it up to the floor quite early on to give you all a chance to ask questions to this amazing panel. So my name's Lisa Melton. I'm a news editor for a journal for Nature Biotechnology. But I started here in the UK. I came from Buenos Aires to do a postdoc at the NIMR in Mill Hill, today's Crick Institute. So this is what I'd like to start with. Someone, one of the stellar scientists at the Crick, yesterday, his name is Sam Rodriguez. He's a spatial transcriptomics scientist. And he posted a tweet that said, how was it? Why do we accept that it takes 15 years from discovery to a drug reaching the market? Have we become complacent? Why does biology take so long to understand the molecular causes of disease? So I thought this is fabulous because I would like to ask this main question: can diversity and diversification help with this particular speed? In fact, there has been an explosion, I suppose, in the last 5-10 years. It's driven by population-wide collection of data. Governments have got behind this. We have the UK Biobank as an example, but many other governments have funded these huge projects. Technology has become cheaper. This is hugely important to be able to sort of obtain the extraordinary amount of data that's coming out of these endeavors. And then, of course, there are the algorithms, machine learning, deep learning, and the very granular tools that have come on board that we heard about this morning from David. So with that, I would like us to keep in mind what type of diversity is it that we need to speed up this route from discovery to market. And now I'd like to each one of our panelists to introduce themselves, and so we kick off the session. Thank you. Shall we start with Christoph at the other end?
All right. So I'm Christoph Potempa, founder and CEO of BrainCures, a startup in the data science space.
I'm Victor Nedova. I'm a senior director at MSD here in the UK. I work in early discovery and biomarkers. My space is data science and machine learning, and previously I worked across multiple pharma and startups, and was academic before.
My name's Rowan Gardner-Still. I'm a co-founder of Precision Life, and you'll all notice I'm not in the naughty seat on this panel.
Thanks. I'm a dinosaur of the first wave of biotech, and have stayed and loved it. And since doing one of the early UK biotech companies, I have kept on helping to founding companies, investing in companies, and have spread my interests away from therapeutics, as well as therapeutics, into this space between data and life sciences, which I think is particularly interesting. I'm now chair of a set of companies on MRC Council, on the board of Cancer Research UK and other things. But I am a dinosaur.
Great. Well, thank you, dinosaurs and all. So what type of diversity is it that we're talking about? So we are now at a point where we can look at geographical population representation, for instance. We can look at diversity in different approaches to target discovery, or, for instance, diversity in how genomics and other type of multiomics are allowing us to match patients in clinical trials, or are we talking about diversity in investment models, perhaps? So, Victor, I'd like to ask you if you could start with, you talked to me about a very interesting project that the UK, again, is spearheading with our Future Health, and why it's important to bring in population diversity.
Yeah, thank you, Lisa. So I do believe that more diverse, high-quality data does provide us with a more important insights into the biology. And we're currently at the cusp of really getting more economical way of accessing large populational cohorts. And you can actually spearheading it with one of this initiative called Our Future Health, which is potentially can be one of the largest cohorts in the world, of 5 million individuals in the UK, where we'll be able to profile and identify markers of disease, not only of disease incidence, but also of disease progression. So this will be very important for the population and for the UK PLC as the whole. And I think hopefully it as well will contribute back into the NHS. So this will will capture the diverse UK population, which is, as you recently probably heard the census, the UK becoming more and more diverse, and it's a very important aspect for, at least for genetics, because you can really capture multiple different insights from using ethnic diversity and both age and gender. So I think those type of cohorts will be really important for identifying new targets, but also finding new biomarkers and the right populations for the clinical trials.
Great, thank you. With that, I'd like to be a little bit provocative and ask Rowan, is diversity always better?
So my reaction is to this, because I'm going to roll in the setup question as well, which is why does it take 15 years and why does it take so long to understand biology? So my answer is I think we're using the wrong tools. Generally, the wrong analytical tools. I'm equally committed to the gap between data and biology to Andy, and I think undoubtedly more diverse data has real value, but only if we're able to extract insights from it, and that we can access that data. So that's kind of part of the corollary on the Our Future Health piece. And in terms of, what kind of diversity do we need to get much better at moving those insights to a place where it can do good and have an impact on patients' lives? I think that's a much wider embracing of what diversity means in the value chain. It's as much about really good business development capability from small companies and large companies to get on board with new technology. It's also a diversity of funding. it's a different mindset to invest in these tech bio companies than it is to follow the therapeutic path. And we have much stronger skills in Europe and world-leading investors in the therapeutic space, and we have less capability along that whole capital cycle that Kate was talking about in the earlier sessions in tech bio in Europe. So there's a whole ecosystem piece around what it means to be to get the diversity working for us.
Thank you. Good thoughts. Christoph, when we chatted earlier, when preparing for the panel, you mentioned that you had great sort of frustration that nothing had changed in the neuro space for a long time. And will diversity help there? And could you tell us about your platform?
Yes. So I've been working in the neuro space for over 20 years. And one of the key reasons I think why drug discovery also takes 15 years or longer and is only successful 1 in 10 times in the clinic is because we don't understand the biology. And that's particularly true in the neuro space. And so with that in mind, my company, one of the foundational things in our company is we decided to go after understanding the biology of long-term learning, or how does the brain learn things. And there's this paradigm of long-term potentiation where you can look at early and middle and late phases of how a memory forms after you have an experience. And we looked at the genomics, and we've been able to identify 3,000 genes, which we then brought down to about 822 targets and built a discovery engine where we understand who are the workers, managers, and directors behind brain biology, and it can help companies now achieve precision medicine, or at least not go on and fail. So we've used the GSK has pooled about 72 drug target pairs that then went on to fail in phase 3. When we used our engine, we were able to eliminate 57 of those failures. So I think that's very powerful, and we need to be applying more of such in silico approaches in the future to make impacts. And I think one of the key things that we need more of diversity is focusing on transitions between health and disease to come on to the concept of scientific wellness. And we need to apply it both in animals and in humans. We need more of, you could say, maybe you call it basic science, but we need to ask the right questions. I think we've got the right technologies. It's now just asking the right questions and doing consortia-led projects that focus on, you could say, this, answering very specific questions of technologies rather than just going for the same target. I mean, in Alzheimer's disease, the A-beta field is theoretically now says it's the success of lecanemab, but I don't see it quite yet there. We've had so much failure that we accept a very marginal change of something that works, where we've got so much other target diversity. But luckily, there's more and more, I guess, target diversity. At least the Alzheimer's Drug Foundation, the US, believes that there's 75% of targets now don't focus on amyloid, which is a good thing.
Yes.
Because I think we've been hitting the same nail with really big failures, and some of these resurrected compounds are resurrected phase 2 phase that then go on to do large cohort studies of nearly 2,000 patients, and we could be much farther along if we didn't have some, you could say, key opinion leaders be in the driving seat, or have more diversity of key opinion leaders across the targets, I would say. I think that's, I think, the key to success across all indications.
Great. So we need to keep a diversity of targets if we're going to move forward.
And opinions, I guess, of the on the table, right? Because you always get a beta showing up if you choose the key opinion leaders who are the founders of that theory. So you could say people who disagree with you to at least have an agreement or disagreement on about where a consensus, reach a consensus point.
So hypothesis agnostic is the.
It's good to be hypothesis agnostic, or at least build another layer to the hypothesis. especially using the multi-omic tools we've got, where we can take a single drop of blood or saliva and get so much insight. Like there was this company, Arivala, set up by Lee Hood in 2015, where they did 108 patients and they followed them over 9 months. They did panels every 3 months, health coaching, and they identified hundreds of biomarkers that then were able to, for I think 3 patients, stop them from going on and developing a disease. So I think these are the kinds of projects we need to do in the future.
Fantastic. I like the tracking idea. So if we're talking about diversifying the approach to drug discovery, Andy, how have things changed?
So as a dinosaur, I'm going to go back into various extinction events, and I can see a number of people here, actually it's wonderful, who were through some of those first extinction events, so bear with me a little bit.
Some of us had better moisturiser.
Because this feels a little bit, in many ways, you could say this feels a little bit like the end of 1997, 98, 99. And a crash occurred at that time. But it followed a period of an enormous investment bubble, and more than anything, incredible certainty about the future and the way it works. There was that diagram, you know the one, you could probably still draw it, George Post, Jürgen Drews, etc., etc. There was great certainty about what the future was. This diagram that said there was, genomics was going to give us a tsunami of new targets. Combinatorial chemistry was going to throw billions of molecules at it. Informatics was going to
I think I drew the diagram.
Yeah. And by 2002, we all knew it hadn't worked. Well, lots of us did. We actually weren't admitting it to the public markets, which was, again, a big shock and one of the reasons why the industry suffered. There was no productivity gain in pipelines, other than perhaps in areas like antibodies, which, by the way, were never on the original diagram. And so we should beware, right? But I'm an optimist, and I think the situation is fundamentally different. The problem at that time was not that some people had a great plan and a vision and linked it with things. It was the fact that everyone had the same plan. There was a huge homogeneity of approach. And the approach was putting everything at the beginning of a small molecule discovery process. And there were some flaws in that, which we could go into, and that's a subject of another talk, and it's interesting to debate. But everyone failed together, largely, and I'm making generalizations here. I think the situation is very different now. We have an enormous period of investment that's gone into the space, and we're going through a bit of a crash. But the thing that's different is the diversity. And that's a diversity across the board. It's a diversity of approaches, of technologies, of modalities, of where it's being applied. You know, yes, it's being discovered it's being applied in targets, but it's also being applied in validation and in discovery and in development and on market and on expanding and selecting patient populations. It's in diagnostics and digital therapeutics and the biomarkers, as well as in therapeutics. And that makes a change, because actually novel diagnostics and the detection changes things. It's the thing that will change everything in some of the neurodegeneration areas, by the way. And it's not just a diversity of, across that value chain, it's a diversity of investors with different business models, of corporates with different business models. There's as much activity from the tech corporates, from Google Ventures, from Intel Ventures, and from the health systems, Optum Ventures, Blue Ventures, of Blue Cross Blue Shield. It's a really interesting mix. And I'm a Darwinist in terms of innovation. I believe in diversity and selection pressure. Now, the result of you've got to have the right diversity and you've got to have the right selection pressure, and too much selection pressure, too little selection pressure, and you end up with a Cambrian explosion of a load of things that are going to die out. Too much selection pressure, and obviously you end up with nothing. One of the implications of this is lots are going to fail, but I think we're not all going to fail together. And I think that's the good thing. I also link the diversity of people very much to this. So I was yesterday, had the real pleasure of hosting the first Cancer Research Horizons Enterprise Awards. Fantastic spread of companies. We were utterly surprised by the number of nominations we got, which was a real pain in terms of a judge who had these zip files arriving on a Friday to judge on the Monday morning. But it was the diversity of modalities, of business models, and of people and their own journey that spread into these things. And I think that's what's really important. So I'm a sort of optimist in this because of the diversity, and it will be really interesting to see how the sort of, you know, the recent contraction in the financing environment is going to create the selection pressure where some things are going to come up and some things aren't. Is that helpful?
Very. The Darwinian perspective, always welcome. Talking about Darwinian, I would just like to take it back a little bit to the technologies. At our journal, of course, we've been tracking all the CRISPR, and this is sort of history now almost, but this really shook up the whole life sciences community. But then we had, for instance, omics of every type coming on board. But the one thing that I feel has caused the most excitement recently is the AlphaFold and the Rosetta Fold. And I just wondered whether any of you would like to riff on this and how this might accelerate or not.
Can I start? Is
Of
that
course.
okay? Because I know you're a specialist, and you're so coming back to my.
I don't look excited, do I? So let's undo it.
It's interesting where it's sort of interesting where it came from. You know, this is DeepMind, and this is Google and DeepMind and Demis deciding to do things that are difficult, and AlphaFold is fantastic, different model. Put the whole thing out there, get people to use it and feed the data in. I mean, there are big flaws in it. You know, if you've got a metal in your structure or a heme or something like that, it doesn't sort of handle it particularly well because that's not in the primary sequence. But it is useful. It is useful, and it would be really interesting to see whether the new company that's been formed as this actually takes a different approach to how it finds its therapeutic modalities that come out from that, because by its nature it's probably going to be small molecules, I would imagine, given where our folders come from. I do think that some of the chemistry approaches are lagging behind the biology approaches in all of this. I think that's one of the things we've got to do. But again, I think it exemplifies what's happening out there at the moment in terms of diversity of approach.
Yeah, there's about a new company in that area every fortnight, as far as I can see. So it's really happening. Victor, did you want to?
Yeah, so maybe if I can add, I think we should not as well get into the trap of discussing one versus another. I think it's a really good to have a more different approaches. I don't think drug discovery is as complex as you can all know. It takes very long period of time, and there's all different types of silos and issues there. So I think diversity of approaches would be really important in every aspect, and not probably one will solve all of it. So I think it's important not overpromise on any of the approach, because it actually will be harmful in the long term.
Yeah, and,
So
and actually we did overpromise in 96, 97, 98, 99 to the investment community. who
Yeah,
then
it's a
decided that pharma-
natural
yeah,
way
yeah.
of this business. But I think it's important when we're applying it, it's to have as well, I like your idea, and to have a multiple approaches. So for example, we are applying assembly of approaches on assembly of the data, just because, I mean, biology, we don't have a great theory of biology. Biology is now like physics was 300 years ago. So we're still kind of waiting for an apple. Apple already probably fall on our head, but it will fall again. So I think we need to develop better understanding of causal biology, and I think we're on the way because of the amount of data and amount of methodologies will help us to uncover more and more important insights. Yeah.
But I think that the biology has to have a context of a disease or indication to be applicational. And also I think one thing I really gets me nervous is when you talk about, let's use a drug for a novel drug for existing indication. I think we should make existing drugs for existing indications. Right? I think there's been too much focus on when we work with a lot of clients, there's always more of a shift that we want something novel. Like, can your platform do something novel? And say, yes, it can, but that's why we're still at the 15 years to get to the patient rather than 5 years, and much more economical because you have a lot of safety data. So I think until someone is able to show that in a more streamlined manner, there will be groupthink still going for the 15 years model because of the intellectual property and just the whole cascade of things we were all quite aware about that lead to, you could say, the monetization of the companies.
Mm-hmm.
When Andy was being he was Dino, the little dinosaur. I worked for a company called, exactly, Wilma here. So I was Wilma at Oxford Molecular, and we had a couple of tools, one of which was written by my husband, which was a relational database of protein structure, right? So I've been interested in structural biology since I opened Carl Tooze's book, right? So it's the most beautiful discipline in the world. And I remember sort of him sort of pouring a glass of wine and wide-eyed and witlessly sort of waxing on about solving the protein folding problem. And come 2002, 2004, I think when he was working with Andy at BioWisdom, he came home and he said, well, there's no bloody point. I'm making it like slightly politer, solving the protein folding problem because it's such a small piece of the jigsaw. So yes, you know, I think what they've done is remarkable. It's interesting. Will it move the needle? I doubt it. And it's to do with we can string all of these things together, but ultimately we have to recognize that biology is combinatorial, it's non-linear, right? And yet we're still overly reliant on the analytical technologies that were being dreamt up when we were working with Jim Watson, it's always a good name to drop, when he was writing the original grant for the human genome back in the 90s, so I'm still Wilma at this point. And you know, we are still interrogating genomic data based on comparing single data points, right? Whether there is a variation or an insertion deletion at one point. And then we find these things and we imagine we can string them together to build a whole complete model of our disease that has explanatory effect. And that will never work, right? Just because you see these variants, they're not necessarily linear and additive, right? Our chronic diseases, where I do most of my work now as Grandma Wilma, is based on patterns, is on interactions of properties, and it needs a different maths. And if you're only putting into your machine learning models insights that you're deriving from single points from data, you're never going to have a complete picture. It doesn't matter how much data you have. And I think that's the challenge that you know, I think is really interesting, but we need much better analytics. And that will find us answers faster. And I'm really excited about what we're doing at Precision Life, because we have underpinning all of the stuff that we do and a mathematical approach. There is a lossless dimensionality reduction so that we can hold combinations of biology together through our analyses. And it's unlocking magical insights into diseases at a speed that I never thought was possible. And I'll just touch on COVID, if I may, because it's the COVID, what we learned in the pandemic, it was a wonderful moment that we can actually benchmark different approaches, to our understanding of disease biology. It was quite a complicated problem. You had both the host and you had the infectious disease piece. And I think it's useful because it kind of begs the question, is more data actually going to be prevail, right? Can we data our way out of this? So Precision Life took the decision that it would put its whole technology at the whim of anyone who wanted to analyse COVID-19 data, and all the results are in the public domain. 23andMe, they had quite an interesting data source. They went and analysed their data. They found one gene, which was nothing to do with anything associated with the mechanism of action at all. So that was a lot of money spent with little value, and you know, the COVID-19 Host Genome Consortium, right? They are our best bioinformaticians globally, and they had access to, I think my last count was 49 different data sets from around the world, right? So across different modalities, genomics, diversity. It took them a year. They found 9 associated diseases on 3 mechanisms of action, so considerably better than 23andMe. You might argue that it's the type of data. So Genuity put a publication out this year. Now, Genuity sort of is the masterclass in multimodal data, boil the ocean. They found 5 genes, or maybe it was 6, associated with one mechanism of action, viral infection. I didn't need to boil the ocean, store the data, right, to look at that patient data and know they had a viral infection because they had a COVID-19 diagnosis. Precision Life on the first 750 patients in the UK Biobank, in the first 3 weeks of that data being available with a combinatorial approach, found 68 genes, candidate genes, across 6 mechanism of actions, which explained 12 genes associated with host immune response to infection, 5 genes associated to the regulation of inflammatory cytokine cascades, 5 genes in lipid storage, signaling and droplet production, 12 genes associated with cardiovascular and endothelial function, 9 genes associated with Wnt and beta-catening, i.e. cell senescence, which was kind of was surprising, and 6 genes associated with neurodegenerative diseases. So pretty much what we were seeing in the clinic. We went back to the analysis in March, and 48 of those genes have been validated by the wider scientific community in the peer-reviewed literature. We also then read from those targets and did what compounds might treat them. 29 were suggested for repurposing. 13 of those have been through the clinic. 1, dutasteride, was just read out in a phase 2 double-blind RCT as being very effective in controlling viral shedding within biology. And we looked then at a second data source of clinical data from United Health Group, and sort of doubling in on the endothelial cell function and the lipid biology, we looked at dyslipidemia and high calcium signaling, and were able to put together a clinical risk score and recapitulate those findings. And that's important because the United Health Group data has a very different genetic ancestry to what's in the UK Biobank. So, you know, we've only published 2 studies, and they're not our best studies by a long shot, but I think it's a call to action from me that we need better analytics to get the value out of this data, and actually to feed the insights into the machine learning models that we're building, because otherwise we're not actually recognizing the signal that's in the biology. And if you can do that, I think it will be transformational, right, to the drug discovery industry, and it would be great because neither of the dinosaurs are getting any younger.
Yeah. So maybe that's a really great insight, and thank you, everyone. I think it's important really to be true with our idea of diversity. So the way we're approaching this in my neck of the woods in MSD, we are trying to get really longitudinal, diverse human data. And I do believe that it's important to get really robust data on a number of diseases, both generated internally and externally. But we're also using, as I mentioned, assembly of approaches. Because we've seen that multiple approaches does bring machine learning and statistical approaches, does bring slightly different lists of targets.
Yeah.
And again, I think they're all lists, they all need to be validated. And there's a very difficult to benchmark those things. Sometimes it's a lot of this, do I think that lipid biology plays a role or don't I think so? A lot of this, it's biology insights, and we know that they are all very biased. So I think it's important to be very true and agnostic when you kind of examine those lists. And I think that, of course, the true validation is in the patients, and which is, again, 15 years to wait, and we cannot afford to do it. So we're trying to make a proxy markers for this. We are looking at this in specific functional phenotypic assays, and we see how many of them are validating. And we're designing a cascade assays, which gives us a bit more robustness to start really a drug discovery program on a specific target. Because if you think about this is where the really churn and investment starts, when you start doing chemistry, when you're putting 20 chemists on a program.
So I agree. I mean, you know, it's really important that we recognise what comes out of what we're doing in silico are just testable hypotheses.
Yes.
Right? That's all they are.
Yeah.
The link between having something that's hypothesis-free and is a testable hypothesis and how quickly it can be tested and validated is the key to having greater confidence in the value of data, the data that we have of it.
Yeah. And there machine learning really can play a role. When we have inputs which we can then put back into the model, we can start iterating on this.
I mean, the challenge back in, the certainty of 97, 98, was we underestimated the complexity of biology, and we're still a very, very long way from understanding biology. It has so many, it's very complex, so many control layers we haven't yet thought about, such diversity at cellular level, at interactions between cells. So it's not an easy problem, and it's not one that we have.
Thanks. I'd like to bring you all back from this in silico reality to the real world biotech industry reality, and so I will revert back to the definition of diversity. That was what I first thought of when Tony approached me, and that was that female representation in UK biotech. This is sort of a little bit of a digression before we open up to Q&A. So the last white paper I saw was 2020 by Liftstream. They reported 18% women in boards of UK biotechs and 7% of ethnic minorities. Is there any way to improve on this within your own ecosystems? Do you have any suggestions?
So I think diversity is important in many different levels. So gender and many other. There's a diversity in age, in everything else, in many different dimensions. So I think it's good we focus on all the dimensions, and of course the gender as well, because it's very quantifiable and it's very easy to fix. But what I think is that we should start early. I think it's sometimes we fall into this things that let's just do it on a specific level, and then there's already very little pool of talent to choose from. So I think what we are trying to do is we try to bring as well children from Camden and try to get them interested in science. And I think it's the sooner we start, the more talent of whatever denomination we will have, the easier it will be. It will be no-brainer to choose from this. And of course, there's but with time, but we all have biases, but they will erode because people will have much more. We will see many more people of different nominations in the industry. There will be it will become transparent, the bias.
Great point. Thank
So
you.
my idea on diversity on boards is essentially, yes, so Victor's, I think the argument is we don't have enough senior people to put on the boards, but hello, we should actually have younger people on the boards as well, and we've got enough younger women who want to take those roles.
Thank you.
Yeah, no, I actually agree with that,
Yeah.
that last point. And I think diversity, you know, my argument, you know, there's a fairness thing, there's all sorts of things, a social thing, but, you know, I actually think it's an intellectually healthy thing, diversity of backgrounds. And clearly, you know, you can put systems into pull. You've got to put systems into, you know, to push and encourage. And that's a long-term project because you've got to have that running through from, you know, different groups. And I would include things like neurodiversity and all of that because I think that's very important because I know neurodiverse people, I mean, heck, I've got ADHD in one way or another. But I know I think some of the skills from people with neurodiverse backgrounds are sometimes really, you know, really contribute, but I mean, one has to have them in environments where
I guess
well,
boards,
so I'm a huge proponent of this, and I think we have to work from the different.
well, sometimes you could think you could have a list of candidates for boards, like the VIA does, sort of, right? When they choose their board and people vote within the company who they want to have leading the company or representing, and at least there should be some dark horses within the company who represent everybody's viewpoint, right? Because I don't think you have employers' viewpoints on the boards often. I guess
So everyone on Team Pink is going to be laughing as the men talk over this piece.
Have a pink pen.
Exactly, you have a pink pen, and I'm just going to lean in and say a couple of things to use the Cheryl, whatever her name was. Firstly, recognise people like Andy Richards, whose investments have disproportionately invested in female-led teams. So he's part of the solution. Right. Secondly, it's just you know, ridiculous for an industry where more than half of our PhDs are female, that we don't have more women at the senior table, around the board table. It's just a really poor reflection on all of us. Because when you have diverse teams, and you're absolutely right, diversity in every dimension, it's really upsetting when I have you know, white male colleagues who are gay, who don't feel able to be themselves. You know, that's not an environment that makes people comfortable, that makes them, you know, do their best work, bring their full power to solving complex problems. And whenever they look at what does it take to be successful in solving complex problems, and I think we've heard today, biology's pretty damn out there. It's kind of complex. It always comes down to a diversity of educational background, of social backgrounds, gender, all of the protective characteristics. And so, you know, what can we do? Well, we can lean in when Kate is on the table saying, you know, I'm running, you know, creating a new fund on investing in female-led. So I'm going to put my hand up and say, how can I help? I took time out today. I'm supposed to be on a call at the moment because I'm president of the jury for the Cartier Women's Initiative. And we award money globally to just incredibly impressive women-led entrepreneurs who are running high-impact businesses. Last year, we gave an award to a woman out of San Diego who's actually got a carbon-positive water treatment technology, right? Sorry, carbon-negative. A very positive impact, Ren, that's the other way around. Carbon-negative water treatment, what that could do in, you know, for sustainable issues is just enormous. And I'm even more impressed with this year's cohort. And I will just take every opportunity to keep reminding you that when you invest in diverse teams, you get faster return on capital, fewer losses, and more successful outcomes. So keep returning that. And my last piece is for all of the men in the room, particularly those who are have capital to allocate to life science ventures is just to keep challenging yourself that you are really seeing a diverse deal flow, because there are so many good businesses out there with diverse teams. And if you'd like to check that, you can call me, because I have binders full of women-led businesses who are chasing capital. So it's a couple of things I would say we can make this industry more diverse.
Oh, fabulous addition. Thank you. Do we have time for? We have 5. Fantastic. Who'd like to ask our panel? Yes, behind red.
Hi there. Thanks very much for an interesting panel discussion. Sorry.
There.
A couple of observations that will lead to a question, I promise. One of the problems of machine learning and deep learning is that they've evolved out of very rich data sources. You know, what data Google mines out of our internet history. And of course, we know in life sciences that that's not the case. Data is sparse. Our data sets maybe contain 1% of all overall data. And the other thing that we haven't really sort of discussed today is the noisiness of the data that we're working with. It's hugely variable. We're often working with models of models of models that eventually lead to somewhere near a patient. So we're talking about, you know, how can we improve R&D output and how do we diversify that? And one of the things that we did better during the COVID period was sharing data amongst companies, and we still work within silos. We're all in our bunkers within industry. Do you think we'll ever get to that point where we might actually start sharing more data to build better models that are predictive in the future, or are we just going to continue in the same vein of hiding all our data?
Yeah, thank you.
So an observation that I remember a conversation with Chris Jones, who used to head up technology transfer at CERN. And he had his last job before he retired was running the end of the Large Hadron Collider to make sure it came in on budget. And he would reflect to me that the particle physicists were very good at sharing data and having large collective efforts, because there was no way you could do particle physics on the bench. You had to collaborate. I think for so long, biology, you could do it as a one-man endeavor with your Gilson pipette. I think what changes that, in part, is seeing some success of the data-driven approaches that we're advocating for, that make the fundamental case that you can get much faster, valuable, patient-relevant insights out of analysing these data. And then it will spin on an axis because people won't be able to share their data fast enough. And we're already seeing that. So at Precision Life, I mentioned earlier, we have an agreement with a top 5 pharma company. We've downloaded their phase 3 data onto our disks to analyse, and we will own the outputs of that and present them back for them. And I think that's a really good example of how quickly people will start to share incredibly valuable commercial data, providing you've got the right information governance stuff in place to drive forward that kind of innovation.
So if I may add, there is a realization that this is this severe problem. And it's even more severe for the startup and for biotechs, and even for players like Amazon or Google. And so personally, MSD is playing an important role in this as well. We are part of several UK and global consortia to generate multiple populational-based data sets, which will eventually become, after some priority period, will become public. And I think there is a more and more of this democratization of the methodologies, and as we're talking about diversity, there's a lot of now high-throughput methods which generate a lot of data. And I think they naturally are more easy to make them more public. So I think the direction of travel is to make it more accessible, but it will be a journey. It will not happen over a day.
I think there has to be a fundamental question behind the data collected, otherwise you're risking to have the garbage in, garbage out output, or no real outcomes.
Okay, one more question. Thank you.
I was wondering the panel's views on technology as a means of democratizing and therefore increasing diversity in R&D outputs. Because yes, X-ray crystallography is a lot better than AlphaFold, but if your company, institute, or even country can't afford or doesn't know how to do X-ray crystallography, AlphaFold's better than nothing.
Yeah, no, I think we just discussed this. If I understand, I think it's definitely the technology will tremendously help not only the countries which don't have an access, but even to the in a large companies. I mean, doing the X-ray for a number of targets is expensive pursuit, and so this will definitely be very welcome.
Great. Do we?
One lady there, question.
Oh, yeah. Can you hang on to get the microphone?
Thank you so much. Thank you. Hi, I have a question in regards to bridging the gap between basically, obviously the research side, but also the clinical workflow side of the sample collection, because I guess that there's a problem also with differences in protocols in terms of like cohort management and sample collection, and also that's kind of like an impediment to unifying data and to having like efficient processes. And I think that there's a scope for technology there in a clinical kind of like workflow management tool to help to unify protocols and better like catalyze like the research process.
Good point.
I wanted to get your opinion.
Yeah. So yes, I think you're right. I think you're right.
Yeah, I think it's important. I think there would be some, and there are some. We are dealing with this problem, and so maybe in a pharma, in a large pharma, there is some more unified way of augmenting the data. But even there are some differences. So it's definitely a very very important when people should get more attention to this one. Yeah.
And I think
I think yes. But there is a very dangerous piece of groupthink here, which is if we can just have a big enough pile of consistent data, we will get the answer. And actually, on the evidence of what we've seen, I mean, excellent publication, Nature Biotechnology. I encourage everyone to subscribe, and you'll find a lot of, for example, GWAS analyses, right? And and it hasn't really changed the game in terms of identifying novel targets or disease risk, except in rare disease. And we're much better at stratifying oncology through genetics. So yes, consistency of data collection, but a real rigor in how you bring data sets together and ideally collect them at enough scale to be useful.
And there are challenges with clinical data sets that are harder than other sets. Understandably so.
One more.
Quick question. Don't you think you're very privileged now? When Andy and I started, we were 64-bit, computers, but you're in such, so we were dealing with blunt tools in those days, which generated billions and billions of dollars worth of drug development. And here you are now, today, with much sharper tools, to actually, move forward in very exciting times.
I think the biggest challenge is going to be the patient engagement, us getting involved in producing personalized medicine.
So that's happening in cystic fibrosis. It's happening in so many areas.
I fully agree. I think there's a very interesting times where we'll generate much more understandable causal biology, and diversity of the tools, as Andy mentioned, will help to generate. So from all this kind of explosion, there will be very interesting new tools coming up, and which will, I believe, will real impact. Yeah, and then more targets coming from populational studies, more insights and biomarkers. So I think it's a very exciting time.
I can tell you were exec, you had 64K. Right? Not all of us did. And so having been on that journey, just tempering that. Right? And so, and I think, it's an incredibly exciting time to be a bioinformatician. I knew if I'd waited long enough, I would eventually get into fashion. But, the reality around... Oh, sorry.
So... Great. So what a fabulous panel. Thank you so much. Please join me in.